Thanks, Cheng. Appreciate the introduction. Great to see you all here. So what I'd like to share with you is the environment in which we do kinetic modeling at the Yale Pet Center, and particularly acknowledge that some of the key individuals who have helped develop this system, Jean-Dominique Galzo, Mika Naganawa, Ansa Homer, and Jongdong Sung with our system manager, for which we would have no data. So quick view of what do we have? We have 4 human PET systems at our Pet Center, the old HRT, the newer MCT, the newest Vision, and the very new Neuro Explorer. We also have 4 small animal PET scanners, 3 Focus 220s, 1 Invion, and we have a unified system for being able to look at all of these data that I'll get to. But what makes our particular place pretty unique is that we do a lot of tracers. So this is the list of every tracer we've done, where we've done more than 10 human scans, and I'll get advanced. I just queried my database for this one, list the number of those scans, and that column labeled FIH says first in human, and to the best of our knowledge, that was the first time that tracer was injected into a human being. So lots and lots of new tracers. So the environment needs to adapt to not doing the standard stuff, doing something new we have to figure out. So when we look at that with our academic requirements there, we want something that is adaptable to new tracers. We want it to be automated so that we can easily process a whole protocol and then reprocess that data when we figure something out, and then reprocess that data when we figure something else out. So we have to have that flexibility there, but at the same time, we need to have it be nice and controlled, so we can validate the code, know exactly what version of code we did, and track everything we've done to the data, each step along the processing pathway. So we have all of our DICOM images are stored on our Linux server. We're dealing with archive and backup appropriately, and we have a lot of custom pipelines really tuned to either tracer or every protocol. We started this a long time ago. The initial package came from NIH, where we were using a package called IDL, an image descriptor language. For those, some of you are used to that one. We can also do this in MATLAB, but this is actually a limitation because this is not a common language anymore. So age does not always have its benefits. We have all the production code in our Git repositories. Only a few select people are allowed to make entries in there, but individual users can write their own code and get it checked in. And the key thing is our database that helps us hook all of this together. We call our database Haven, and I'll give you some really good detailed examples of that, because that's the key that glues all this together. So a typical study, it's long and tedious. We put in the subject, we start the scanner, we inject usually over a minute. Many studies with arterial blood samples, so that's all that ancillary data that we have to use. A lot of them with metabolites, more ancillary data. Do the dynamic scans, and then reconstruct, and then begin to deal with what the processing was. So what's a normal processing pipeline look like? Many steps. So we might take the raw HPLC measurements and turn them into parent fractions, fit that with a line. We'll take the PET images, if it's a brain, register them to the MR. We might take that into an atlas, either FreeSurfer for subject-specific or a generalized brain atlas, generate region time activity curves. If one of those is a reference region, we'll fit that tail to get rid of the noise. We might fit the early portion of the TAC to be able to measure a time delay, so all these sequence of steps. And finally, we get to the meat, taking either regions of interest or voxels, if we're doing it parametrically, and apply that to the relevant set of models we're exploring for a given tracer. We might weight the data based on the counts, which we might get from the DICOM headers. We might have to pre-smooth images, et cetera. And then we have to save everything we've done, which parameters we've used, all of those parameters, so we can go through each of those steps. And all of that can be saved in a way where we can do QC on that to validate, did the motion correction fail? Was there a problem in the metabolite correction? So what algorithms do we have in mind? So when we do this voxel base, obviously one tissue compartment, pretty much all these can do with or without a blood volume correction, fitted or fixed, assuming we have that. Some of the multilinear methods, MA1 and LEGA, 2-tier reversible voxel by voxel, SRTM, MRTM, and the 2-parameter versions. The PATLAC versions, either in its original PATLAC form or its multilinear form, and the generalized PATLAC with a K-loss. Regions of interest, we can do all of those, especially now the more complicated. 2-tissue compartment with 4 parameters, and Logan models with original and reference. All of those together can be done very easily to take the same data and apply it with many different models as appropriate. But the glue for this whole thing is our database. And this was something we had started at NIH. I was able to bring that to Yale, and it has grown tremendously over that. Underneath, it's an SQL database. And basically, it's got 2 basic parts. It's got all the primary data, all the scan data, all the injection information, synthesis information, series information, et cetera. But then what's neat is it has these unstructured tables. For every new analysis, we can create new fields And they're not stored in their own individual tables, they're stored in generic tables, and we have tools in the database to extract them well. So we can look up every injection that we have ever done with great details. I'll give you examples. We can pull out the time activity curves, ROI values, all those ancillary informations like plasma free fractions. And every step of the processing is tracked. We know who did it, and we know when it was done. Users can create queries to get that information out, so our clinical users can be taught how to extract region of interest values and dump them easily to be imported into Excel or statistical packages. But we also have to control who has access to given data based on the user and what protocols they're permitted to do. So to date, there are, I looked it up, over 18,000 injections are in this system in all species, from mouse through bird, through monkey to human. So here's what the database looks like. I'm going to meticulously go through every element in this field, so please pay, no. So the idea here is there's a lot of structured fields, if you can look at it, which this is obviously too small to see. Everything from information about synthesis, information about protocols, and more important things about information about series, that is a set of images, etc. But also these unstructured tables. Maybe we're pointing to a file, or maybe we're just pointing to a single number or a string, or a blob, database word for what could be a curve or any structure in being able to do that. And this database structure has evolved over time as we have added more fields. So how does that affect our pipeline? Well, the idea is that each of those steps I showed is all talking back and forth to Haven. So if I wanted to fit the parent fraction, I'd go to the database, get the parent fraction ones, generate the fit, which would be custom for that tracer, put the fitted curve back into Haven. And then when I want to do the modeling, I would pull that curve from Haven, generate the fitted values, put the results back into Haven. So a lot of this is back and forth. All right, so let me just show you a few things that we can do just to get a feel of the level of detail that is there that has helped us to be able to handle all these particular cases. So I did a query there to say, show me every human UCBJ scan in 2022. This goes off the bottom of the screen. And we get everything from a patient ID that's anonymized, unique IDs for the injections, the injection time, the injection dose, what protocol, that's a code for that, who's the PI, what's the state of the subject, et cetera. All that's available at our fingertips. That's been a great advantage when we wanted to pool data across different protocols, for example, looking at age and BMI effects on healthy control data with appropriate IRB permissions. Here's the example, and I appreciate all these are too small, but looking at the level of information. So for every injection that we do, not only do we know how much injected activity and what time down to the second, we know how much the pre-assay, the post-assay was done. We know the specific activity at time of injection, what the mass was done. We even have the idea of who drew up the dose, able to do that. And the chemists are involved too, that everything they've done about the synthesis is there. We know the end of bombardment time, end of synthesis time, all of the steps they made about doing pre and post injection measurements there. Their measurement of the half-life, that's required for real, so that the 20.4 minute half-life for carbon-11 came out 20.3 in this case, and down to all of the subtleties of the amount of impurities and the legalities of there. So here I did a query associated with, for a given injection, what is the kind of information that might be there? So for example, there might be the curves, the whole blood and plasma curves. That's the raw data, becquerels per ml, decay corrected to injection time. I know exactly who processed it and when. The HPLC measurements, that would mean here's the 3-minute post-injection using our column switching method. I have or down here where we might be having the free fraction, an additional piece of information that was stored, or even information about the injection, the pre, the post-assay, all of there. This is great when something looks a little weird, you can go back and you're not having to have people go through logbooks to find it. It's all already on the system there. For every single set of blood samples that we count, we have a ridiculous amount of information about them. We know what the decay corrections would be. That is the midpoint of counting time. We know what the sensitivity that we applied. We know what the dead time correction that we applied. And all that stuff is automatically stored there. Code written once, used thousands of times, so that's automatically there. How often do we use it? Not too often, except when something's a little bit unusual. Maybe a particular bad point in a curve, maybe the volume was small for that one. If I want to look at the images associated with a given injection, those series, here's an example where for a dynamic scan, this is the UCBJ, it has, all of our tracers have a different internal code, APP311 is UCBJ, and this was the first injection that day, VC stands for Vicra corrected, that has that dynamic scan. We processed that pixel by pixel. We created a K1 image, and was a VT image, which was called DV, for either 60 minutes or 90 minutes post-injection. Or maybe we're using SRTM2, we got R1 images and BPND images, here using Centrum Semi Valley as the reference region. Many, many reanalyses that can be done, different versions, different tunings of parameters. And about each of those series, we know when the scan started, we know all the coordinates, the dimensions of the information are there, and we even know where the file is stored, so you don't have to know where the file is stored to access it, which is great for a system manager. If we get a new disk and they rearrange, they update the database, it doesn't affect anybody doing that. And when we create a parametric image, all the parameters we use, so if I'm doing a basis function method and I have a range of the eigenvalues you're using, we can define that there. We know what model we've used, we know what time shift we use, what time period we've done. And finally is, I think the last example, just being able to look like when once we've generated those parametric images, I know how I created them, and then I can apply regions of interest, extract their values, VTs or K1s, or perhaps I can be looking multiple regions, maybe with a gray matter mass or coming from a CAT segmentation. So a tremendous amount of power. That's the good news. So it's very powerful, it's very flexible, it's great when we're trying to do something new. It is complicated. The learning curve is steep. It is not as compatible with the newer tools. We need a Python interface. We need a much better MATLAB interface. And that requires the F word, funding, okay? And that's a hard thing to get for this kind of work. We've been able to put that together. We don't have enough for the documentation. So it's great for an academic environment, but it looks a little bit like that kid's room that Global just showed in the previous slide. I should have included that there. So that's where we stand. Very powerful. When we're used to using it, we can do a lot with it, but it's something where we're the challenge of being able to use that complicated environment in a practical way still rears its head every single day. Thank you.